Hello, my dear students. Wish all of you are fine. And I'm your course teacher, Ifat Jahan. Today, I'm going to discuss about the reproduction of bacteria. In our last class, we already read about what is bacteria, the structure and classification of bacteria. And you have also finished your third quiz on that. Today, we will read about actually the reproduction process of bacteria, and here we will read how bacteria can reproduce by asexually and sexually. So we know first that what is reproduction, as we already finished our bacteria structure classification with that. We also finished the viral reproduction, our fungal reproduction. So we know that what is reproduction. Actually, it means to produce or making a copy, okay, to produce a copy or gene or the alike materials of the mother one. Okay, that means when the mother or parents bacteria produce the identical one, then we call that is a reproduction process. In case of bacterial reproduction, bacteria can reproduce by both by sexually and asexual reproduction. In case of sexual reproduction, it happens by two methods or three methods, that is conjugation, transduction, and transformation. And in asexual reproduction, it has lots of process within them. We read someone, and in our viral reproduction, actually we read all of them without the binary fission. Okay, we will read that the binary fission. Without that, the budding, the fragmentation, spore formation, vegetative reproduction, we already read that and we have more idea about it. And the process is actually same as virus. In case of bacteria, there are some changes. We will read that. So first of all, we will start our conjugation process. So if you go for the meaning of conjugation, actually what is the meaning of that? Conjugation means that will be the conjugate with something, okay, or it will add with something. So if you go to the figure, that is, it is a bacterial cell and it is also bacterial cell. Actually, in conjugation process, two bacterial cell come together, and there one is actually donor cell, that is cell as say F plus, and another is recipient cell, that is called F minus. There is a changes between them, that in donor cell actually plasmid is present, but in recipient cell plasmid is always absent. Okay, so what happened is, how it's happened, that this recipient, this receiver or the donor cell, actually this donor cell added with that receiver cell, and it form a bridge which is called pilus. Okay, it's form a bridge pilus which is contained by the donor cell, and through this pilus one fragmented portion of plasmid actually transferred to the donor cell, and after transformation that pilus actually is broken down. Okay, that pilus is broken down and it produces two kinds of cell, and new cell is actually happen as a donor cell. Here plasmid will synthesize it and it produces a total mature fragmented DNA or plasmid as the donor cell and with a pilus. So after finishing the conjugation process, the donor cell actually produces an identical another donor cell, which is then again attached with another recipient and can give that plasmid. Okay, so it is called the conjugation process of bacterial reproduction. Then our another one is transduction process. This transduction process actually a process which is recombination. Actually, it exchange of bacterial DNA through bacteriophage. And bacteriophage we already know, it is the virus that infect bacteria. This transduction can be two types. One is generalized and another is specialized. In case of generalized transduction process, it is actually happen by donor bacterial gene is transferred, and in specialized cell it's actually certain donor genes can be transferred. So how it's happened, actually it is very familiar to you, that you already read in your viral reproduction. Okay, that is the lytic cell. Okay, the lytic reproduction, at that time we read like lytic. Now it is the generalized transduction steps in bacteria. So as like that, first of all, viral genome will introduce within the bacterial cell. Then these viral DNA is and the bacterial DNA will be synthesized or broken down in different part, and that synthesize a mature DNA. Some virus will carry the bacterial DNA, some virus will carry the viral DNA. Okay, and after that the cell will be lysis. Okay, the cell will be lysis, and then the bacteria will be removed, or you can say released from the bacterial cell, and it will be destroyed. Okay, and after that what happened in that transaction process, that is the chromosomal DNA, viral DNA just injected DNA genome. With that, then it's synthesized, and after that it's synthesized like that, and whenever the bacterial cell is actually lysis, some virus contain the bacterial DNA, some virus contain the viral DNA. Okay, and what happened after that, that DNA that contain the bacterial DNA virus, it again attack another bacteria, and it inject with the bacterial cell, and now it is a bacterial DNA and it is also a bacterial DNA. So what happens, these two DNA will be attached together and produce a homologous recombination, new varieties of bacteria or bacterial DNA. It is happen actually about general transduction process. Then if you go for specialist transduction process, actually what happened there? Here what happened, if you go for, it's actually divided in every step for your better understanding. If you go for that step, then I again go for the last steps, that what happened actually specialist transduction, that before the general transaction, a viral DNA inject within the bacteria, then this as lysogenic cycle. Okay, as you already know about lysogenic cycle of virus, that is that DNA will attach with the bacteria cell and prepare a phase. Okay, actually this DNA, one kind of bacterial DNA that is contained by that virus. Okay, so this DNA will conjugate together and prepare a phase, and in third stage what happened, this prophase actually degenerate. That means it's separated from the bacterial DNA. But whenever it's separated from the bacterial DNA, it just keep a small part of bacterial DNA and produce a new kind of genome. Okay, and then replication as synthesized happen will be down there. That is, it's synthesized or broken down in small fragmented part, and this bacterial DNA also actually fragmented in different parts. Then with that, some virus contain this genome, some virus contain this genome. Okay, and they're released by destroying that bacterial cell or lysis that bacterial cell. That's said there. That again, that bacteria DNA containing virus attack another kinds of bacteria, and whenever it attack that, it actually inject their genome, and now what happened, this bacterial genome that is present in that virus, it actually attach with that bacterial DNA. Okay, and whenever it happen, it just produce a new kinds of bacteria, or you can say that it's a stable gene transfer into recipient cell chromosome. That means if you go from the first, that is a viral DNA that is inject in the bacteria, and then it just attach with that bacterial DNA, and then that process degenerate, and whenever it degenerate, these pick a small part of bacterial DNA and it's synthesized. After synthesization, what happened? It will contain like this kinds of structure, is assembled like virus, and it lyses the cell and broken down, and then it come out in the environment. Then these viral containing virus, viral and bacterial recombination containing virus actually attack another kinds of bacteria. Then this bacterial DNA is attached with the bacterial DNA, and it's again transformed into a viral DNA. Okay, and then it produces new kinds of bacteria. So it's called the specialized transduction process. And this actually even there step by step, that first the phase genome is injected there, then it attached with that prophase and prepare that prophase. After that you can check that that prophase actually uh, degenerate from that bacterial DNA with a small fragmented part of bacterial DNA. Then it again lysis, and after that it again inject this DNA with the another recipient bacterium nucleate, and then it attached to the bacterial cell and it produces new kinds of bacterial genome. Okay, so that is our two kinds of transduction process, and we also read conjugation process. And now we'll read the transformation. What happened here? Actually, in case of transformation process, what happened? The bacteria binds the DNA and transport it across the bacterial cell membrane. Okay, it's called the transform. That how happened that? That is, it happened actually in two kinds. One is with DNA fragments, another is with a plasmid. Okay, whenever it's DNA, fragmented DNA that is attack a bacterial chromosome and is just inject within the bacteria, and then these bacteria can be worked in two kinds. Okay, how it is? One is like that, it integrated within the bacterial cell and it's called the stable transformation. Okay, that whenever it's regenerate or synthesize, it will regenerate like with that part of that stable part of the another fragmented DNA. Okay, so it's called the stable transformation. But sometimes it's just check there, that is it cannot attach with the bacterial DNA, and then it is actually fragmented or it's just synthesized in small part. Okay, and it's called the unsuccessful transformation. That means whenever that DNA fragment attached with the bacterial chromosome, we'll say that it's stable transformation. Whenever it is not or it cannot attach with the bacterial DNA, we will say that it is an unsuccessful transformation. Okay, and another transformation happened with the plasmid. And what is that? That DNA plasmid actually attached with the bacterial cell, and then that plasmid is uptake by the cell. It's called the stable transformation. So transformation of DNA can be, or transformation of bacterial cell can be happen in these two processes. So we already read about the three sexual reproduction of bacteria. So what is the difference among three? Okay, first of all, the method of DNA transfer. In case of transformation across cell wall and cell membrane of recipient, conjugation through a conjugation bridge as a pilus, and transduction it's happened by a virus. Okay, that means in case of transformation, the DNA transfer happened within the cell wall, okay, or the cell membrane, conjugation within the pilus or a bridge, or transduction within, by a virus. In case of plasmid, it transform in transformation, in conjugation, but its transduction process we already read that actually plasmid is not needed in transduction process. In case of chromosomal transform, actually in that case transformation chromosome is not transferred, in transduction chromosome is not transferred, but in conjugation process is checked this sometimes it's just transferred in conjugation process. Okay, so that was our sexual reproduction, and now that is our asexual reproduction of bacteria. In case of asexual reproduction, actually we will read three, that is binary, then we will also read budding, then fragmentation. The main three happen mostly, we will read now. So in case of asexual reproduction, mostly we check that bacteria reproduce by binary fission. Okay, that is through this process. So what is the binary fission? Actually, in case of binary fission, the parent bacteria, as we know, it is asexual reproduction. It is always like that. The parent will be one. So what happened there? In case of that, the DNA or the parent cell will produce an identical one. Okay, So that is binary call. That means one bacteria can produce two, and this bacteria again produce two, this bacteria again produce two identical cell. Then if that is from there, the bacteria from two, the four will be produced. Then these four again, eight will be produced. So it's actually increases by number of two. So it's called the binary fission. So what happened there? In case of DNA replication, actually the cell is elongation, okay, and then a septate is formation between the cell, then it is divided into two. It's very simple. That means your chromosome will be divided with the cell wall, and then the cell, with that chromosomal part, your bacteria will be divided in part to the identical part of mother. Okay, so it's called the binary fission, that is produced from one parent to two identical cell, then two to four, four to eight, like that. Okay, and that is the binary fission actually. Then our second one is budding. You already know that we read it in bacteria, that in budding process what happened? First, a bud is begin to form from the parent cell. Then nucleus copies to the bud. Then it's generally day by day it just transform a new daughter cell, and from that daughter cell, lots of daughter cell can be produced, and it can elongated by chain or it can be separated from the mother. And our last one is a fragmentation. It's actually called fragmentation. Why? Because it's divided. The body is divided into fragmented part, and each fragment can produce a identical one or a daughter bacteria. So it's called the fragmentation part. Okay. So it is the parent planaria, and that is fragmented parts of planaria, and each of the part can produce a new daughter cell. Okay. So it's, we call say that it is a fragmentation, asexual reproduction of bacteria. So we read in our class actually our reproduction process of bacteria. I think you were clear enough. If you're not, then we will discuss it in our next class. Thank you.